Hi everyone, my name is Enoc Martínez from the Technical University of Catalonia, and I'm going to show our work in fish detection and classification at EMSO OBSEA. So in this presentation, we're going to cover the ecosystem monitoring. Then I will go through the object detection in a nutshell, a very brief resume of what's that. So how can we prepare a data set? How can we train our model, what metrics to look at, and how we can fine-tune it. Then I will show some results of our research, and I will finish with some conclusions. First of all, what is EMSO ERIC, this word in the title? So EMSO stands for European Multidisciplinary Seafloor and Water Column Observatory. It's a distributed research infrastructure. We have 15 nodes in European waters, some deep sea, some coastal nodes. And it's mainly devoted to fixed point ocean monitoring. We monitor all kinds of parameters in different scientific domains, such as physical oceanography, geochemistry, and specifically this talk is going to be focused on biological monitoring. And I'm working at OBSEA, which is one of the nodes. And OBSEA is a cabled observatory, underwater cabled observatory. It's in the coast of Spain and the northwestern Mediterranean Sea. And it's deployed in shallow water, so it's only 20 meters of depth. In this case, this is important because we have sunlight. As you know, if you go deeper, you don't have any sunlight, so you need artificial lights. But in our case, we're working just with the sunlight. And it's a multi-parametric observatory, meaning that we have all kinds of different sensors measuring from currents to temperature, chemical properties of water. And we are also taking pictures of the underwater environment, so we can do ecological monitoring of species based on the pictures. So we have been doing ecosystem monitoring for a while now. Over the years, we deployed many cameras, some low-resolution cameras in the past. Now we have 4K cameras, which are much better. We have an archive of pictures since 2011, and we have been involved in some fish abundance and behavior studies. And traditionally, we did these studies by manually analyzing underwater pictures, which is a time-consuming task and extremely repetitive. You can see here some pictures taken by a low-resolution camera that we had in the past. So in the bottom right, you can see how this observatory looks like. We have this like metal thing is the observatory, and we have an underwater camera pointing to this artificial reef where fishes usually hide and play around. So how did we use traditional ecological monitoring? So traditionally, we took underwater pictures. We had a camera deployed there, so we have a cable to the shore, so we can have access to real time to the pictures. This for sure is not it's not common, or not all the cases are like this, but in our case, we could take pictures as we wanted. So we took a picture every 30 minutes, and then a biologist downloaded the pictures and manually counted the fish and annotated all the different species in an Excel spreadsheet. And after thousands of analyzed images, we could produce abundance time series, meaning specimens over time. So and then we could start doing some science. For sure, this approach is very time consuming. It's not reproducible because since you are counting fish manually, maybe you disregard some of the fish, or maybe you do mistakes. Additionally, you require a lot of expertise in biology because it's not easy to identify species. For instance, I have electronical background, so for me, all fish look the same. So you really need experts to say which kind of species it is, and it's very repetitive, extremely repetitive. Imagine that you have to count fish in, let's say, 20,000 images. So now we are moving towards an artificial intelligence-based ecological monitoring. So we have the same pictures, but instead of counting fish one by one, what we do is we train an object detection model, and then we just throw all these pictures to this model, and the model returns the predictions. And then with these predictions, instead of using an Excel spreadsheet, we store them in a proper database, and then we can extract all this abundant information from the database. If we compare the two approaches, the deep learning based ecosystem monitoring has some drawbacks. For instance, you require some IT skills for sure. You need significant effort to set up the system, but then it's fully automated, and you don't require any human intervention, and you continually get your data, your abundance data, without having to work on it. So, and this allows us scientists to do science instead of counting fish, because biologists spend a lot of time counting fish one by one, and they should spend this time doing actual science. So how can we do this object detection? So a very short and object detection in a nutshell, as I call it. So it's 6 steps. First, you need to collect some pictures. If you want to, it's important that if it's very difficult to have a model that is able to do everything. So we focused on our area. We have a lot of images from our area. We more or less know the species that are there. So we took a data set. Then we labeled our data. So we took some of our images and manually just labeled them. So this is a squid, this is a sardine, whatever. So we labeled all our data. And then this is a tricky part, but you need to select a model. There are a lot of models out there, and it's very mature community. There are tons of different models, and to be honest, there are too many of them. But anyway, somehow you have to select a model, and then of course you need to format your data, because every model expects data in a slightly different way. So for sure you will have images, for sure you will have labels, but some of them are labeled as TXT, some are as XML, whatever. So you need to just convert your data to what the model is expecting. And then you have to train your model with your data. And finally, if everything is okay, you can start using inference, so predictions with your model, and you will get something like this. So we have a picture here, and the model is telling that we have all these fish, and it is able to classify all of them, or at least some of them. Okay, this is all the steps required to do object detection in an underwater environment. How can you prepare a data set? First, you need to select a labeling tool. So, of course, you need some images, and then you need to label them. So, say this is a fish, this is not a fish. There are a lot of great tools out there. Most of them are open source. Some of them are like freeware, but it's a very mature community. So some of them are Label Image, RoboFlow, Beagle, Label Image, Label Studio. So there's a huge community. A big issue that we have in underwater pictures is biofouling. So this is actually this is an underwater picture where you can see that there is a lot of things attached to the optics, and of course this is difficult because your model is going to confuse this dirt in the optics with some fish. So you need to really take care and try to get as cleaner pictures as you can. And this is not always easy in underwater environments. So this is something that you need to pay really attention to. And finally, one thing that may seem trivial, but it's actually very important, is to label everything. Imagine that we have this picture here in the bottom right, and then we want to label this fish. Okay, this is clearly I think this is an dentex. Okay, you label it, and then it's ready to be used. But wait, because here there are a lot of different fish. Maybe they are not as clear as the first one, but if you are not labeling them, the model is going to understand that this is actually a background. And you want to avoid that. You don't want to cheat on your model, because if you don't label everything, you are implicitly saying that this is background, and then you will run in all sorts of troubles. So labeling, as you can see, is very time consuming. Next thing that you need to pay attention to is class balancing. So class balancing means that you have more or less similar number of labels for all your classes. Classes in this case mean fish species. So if you want to detect cats and dogs, it's obvious that you cannot have 100 pictures of cats and only 2 of dogs. You need more or less the same. But this may be difficult. For instance, take a look at this picture here. So even if we are only interested in the yellow ones, these zebra-like fish, we need to label all of them. And as you can see, that the right diagram is our data set. As you can see, the chromis, which is this small fish, is completely overrepresented. But it's very difficult to get rid of that, because in our environment, they are really common. So even if you have a picture where you want to select some different fish that are underrepresented, maybe you have 2 labels of underrepresented fishes and 20 of overrepresented ones. So this overrepresentation issue may be unavoidable, and it's something very difficult in underwater environments. And of course, as you can see here, this is our data set. We have like 1,700 images. And as you can see, we have more than 8,000 labels from this chromis. It's a small black fish. But there are tons of other species that are very uncommon. This is going to be really a problem when you train your model, because you cannot train with only 10 specimens of, I don't know, apagon and 8,000 of chromis. So you have to do a kind of Solomonic decision here and say, okay, I'm going to include only these classes and I'm going to exclude this from my dataset. So this is what we did. We decided to include only the 12 most prominent species and to exclude the others. Of course, this is not ideal, but it's a pragmatic solution, because otherwise the underrepresented classes are going to be really, really, really bad in predictions. Then once you have your dataset, you need to select a model. So as you know, deep learning is a very mature field, it's rapidly evolving, and there are a lot of open source tools out there. So what I would suggest, if you have to start from scratch, is to go to some sort of benchmark webpage where they benchmark different models. A great one is Papers with Code. So here you can select your application, for instance, object detection, and then it gives you the latest models with benchmark with a standardized data set, and you can compare them. So it's not a trivial task to select one, and this kind of benchmarking tools help you to decide. In our case, we chose Euro-BioImaging because there's a good balance between precision and speed. It's quite easy to use, and there is good documentation available. So now we need to train our data. So as I said before, then first we need to format the data. We have to read the documentation and see how this model is expecting the data, if the labels are in TXT, are in XML, whatever. So you just read the documentation and prepare your data. Some tools like RoboFlow even help you transforming automatically from one format to other. So this is also useful. And then you need to train your model. So, and how can you train your model with your data? Then again, you just go to the documentation, and this is just a screenshot from Euro-BioImaging docs. And with this simple command, just one command, it trains the model. If you don't have issues in the data and everything is as expected, then you get your model, and then you can start doing inference. So you can then you just throw some pictures to the model, and you get some predictions like this. Okay, great. So you have a model, and now it's working, but how good is this model? Then is that we need to measure how good is it. So then we enter in the realm of metrics. So it's boring stuff. It's very specific for deep learning, but we really need to understand it if we want to improve our model. So first thing that we need to understand, the first metric that we need to look at is the intersection over union. So if we have a fish like this, and we have a prediction, then what we need to define first is the intersection over union, which is the common area between the label and the prediction. Usually, if a prediction has an IOU less than 0.5, it's considered as a bad prediction. And if it has greater than 0.5, it's considered a good prediction. This 0.5 is adjustable, and it can be 0.5. Some people are using 0.7, whatever. It really depends on how precise your localization needs to be. Then we have the precision and recall. The precision is the true positives over the sum of true positives and false positives, and the recall is the true positives over the sum of true positives and false negatives. So here we can see an example. So in this case, we have precision of 75% because we have 3 true positives and 1 false positive, but we have a recall of 100% because we don't have any false negative. So as you can see, there is not a metric that covers it all. And in this other case, it's the opposite. So our precision is 100% because our predictions are correct, but we are missing some of them. That's why we get lower score in recall. And finally, we get to the average precision, which is the most popular metric, I would say, and at least is the one that we are using to compare our models. So the average precision is the area under the precision recall curve. So you can, when you train your model, you get some graphs like this. And then for every class, you get this PR curve, and the area below it is the average precision, which is actually a mix of the average and of the precision and the recall of your model. And the most standard way to compare this kind of models is to use the MAP, which is the mean average precision, which is the average precision of all your classes. In this case, we are using the MAP at 0.5, because of course, if you change the IOU, this graph will change as well. So it's a very common metric. I would say it's a de facto standard, and in these applications. And this is the metric that we are using to understand how good is our model. Okay, so now that we understand the metrics, how can we fine-tune the model? In our application, the first thing that we need to tune is the image size. We detected that for speed purposes, most of models usually reduce the image quality when you throw an image to the model. And in our case, this is really a bad idea, because then you are not going to be able to detect small fish. As you can see here, we have the same picture with different resolutions, and of course, the model is going to work better with the first one than with the third one. Of course, this is going to be much slower, but again, if you want to be able to detect small fish, it's really useful. Epochs means how many times the algorithm is going through your data. The higher the better, up to a point. So usually the sweet spot is around 100, 200, depending on your model and your data. Then we also have this batch size, which means how many pictures is the model using at the same time when you are training it. Usually the higher the better, but it means that you need more memory and more GPU. So you need resources. And the best way for sure to improve a model is to have more training data. More training data. So training data is hard to get because you need to manually label the data. So it's very time consuming. How can we speed it? How can we speed this task? So to speed this task, you can first collect more pictures and then do some kind of semi-automatic labeling, meaning you run. So since you have a model, even if it's not perfect, you run some inference, and then you can drop this inference into your labeling software, and your software can automatically recognize these labels, and you can then adjust manually to make them perfect. Then you can retrain your model, and you get this improved model, and then you can close this loop. So in this way, you can get much more data much faster. And now let me show you some results. This is a video inference with a YOLO DA, and with only 400 pictures. So it's not that much data actually, and it's quite good. We had 400 pictures with 12 classes. And actually, it looks great. Okay. Here we have another. This is a bit better. This is not only with 400, since we closed the loop and we did some more data. This is with 1,600 pictures. And as you can see, the small fish are also detected. And you can see here that the big fish are predators, the small one are prey. So it's easy to see the interaction between them. Okay. Of course, once you have these predictions, then you need to use it. It's okay, it's nice to see a video with the boxes, but it's not enough. And what we do then is to extract this abundancy and to create this time series. And then we can use it for, I don't know, any kind of research, predator-prey interaction, correlation with environmental data, whatever. Just as a note, in the past, to analyze 30,000 images, it took more than a year manually. And with this algorithm, it took 3 hours to do it in 50,000 images. So this really, really helps, and this really improves your scientific pipeline. Just some comments about the EImaging project. So this work has been funded by the EImaging project, and we are using this platform, which is quite nice, where you have a lot of different off-the-shelf artificial intelligence modules. You have resources for training, but this is only for people within the project. But anyway, it's worth to take a look. Everything is open source. Everything is in GitHub. All the models are also containerized, and they can be downloaded from Docker Hub. And I think that it's a very good starting point if you want to deploy a model. And to finish, some conclusions. So I've shown a very specific application. So if you want to apply this, you need to first think thoroughly about your application. Is this going to fit your needs? Is this model of way of working going to be useful for you? Then there are a lot of open source models, very mature, very easy to use. And in our experience, quantity and quality of training is crucial. So you can play around with all the different parameters in the training, but the most important stuff is the quality and quantity of your data. And of course, you require some coding skills, especially Python is really handy when you're doing this work. And as a final thought, AI models are easy to use. So I've been working with this maybe 6 months only. So they are really easy to use, but they are not easy to master. So it's easy to have some sort of workflow going on, but if you really want to make the best of it, it requires some time. So thank you for your attention, and I will be happy to answer to your questions.